Hello, and thanks for joining me here today. My name is Dr. Emily May Armstrong, and I'm a postdoctoral researcher at the University of Glasgow. Today, I'm going to be talking about what it means to be a disabled researcher, try and help people understand the basis of genetic disease, and also touch a little bit on why I study the genetic basis of why and how plants grow. So I'm going to start by talking a little bit about what it's like to be disabled in the UK. There are 22% of our general population who is of working age are disabled. But of that, we're missing 3% of disabled people who can't work due to their disability, leaving us with 19% of people who are both disabled and working. But on average, it costs a disabled person an extra £583 per month as a direct result of their disability. That wouldn't be so bad, but on average, disabled people only receive an extra £328 from the government. That leaves us an average of £255 short each month. To make things even more complicated, 33% of the general population view disabled people as less productive than their able-bodied counterparts. And this is pretty bad news for disabled people because not only do we not have enough money on average to live off, but people we work with might view us as less productive. So now I've given you an overview of what it's like to be disabled in the UK, I'm going to talk about my own disability and how my disability affects me. So my disability, my genetic disease is called Ehlers-Danlos syndrome, and it is a genetic disease causing dislocations, chronic pain, fatigue, and innumerable secondary diseases. And I'll go into this in more detail later, but it took me an awful long time to get diagnosed because not a lot is known about Ehlers-Danlos syndrome, and it's quite a rare disease. So let's get back to basics for a minute and talk about the basis of genetic disease. So genes encoded in our DNA, genes are transcribed to RNA, RNA is translated to protein, and if that gene is mutated, you can end up with a faulty protein, and this ultimately can cause disease symptoms in some cases. So let's take a look at how we actually identify a disease-causing variant of a gene. Here's our DNA, and we can sequence our DNA to try and find the mutations that might be causing a disease. Genetic sequencing is becoming cheaper and more widespread, especially in NHS facilities. So what was once a really expensive, unobtainable thing of the past is now a pretty accessible healthcare diagnostic tool. So we pull out the sequence that we're looking for. You can see here that we have 4 bases. They're called base pairs, A, T, C, and G. Now, A and T pair together across the DNA backbone that you can see, and C and G do the same. So sequencing allows us to find the specific sequence or order of DNA bases in a gene. So if we want to find out if this is a problem for the person that we're looking at, we will compare this to our known healthy person's gene or our reference genome. And as you can see, this specific mutation, it doesn't really seem to be an overlap, but when we compare it to the known disease-causing gene, we can see that it's a match. We can confirm that this faulty gene is causing the disease. Once we've confirmed the faulty gene is causing the disease, then we can move on to treatment. And it's important to underline that this faulty gene may have been inherited from a parent, but the parent might not have disease symptoms. And this is because of different inheritance patterns. You can have a dominantly inherited gene where you just need 1 copy, or you can have a recessively inherited gene where you need 2 copies, 1 from each parent, to give the phenotype, the disease phenotype, from the disease genotype. So the phenotype is what the disease looks like. The genotype is the underlying sequence changes that cause that disease phenotype. Bad news for me is that Ehlers-Danlos is dominantly inherited. So whilst neither of my parents necessarily had symptoms, one of them passed this gene on to me. But what happens if we can't find out which gene mutations cause a disease? It can be pretty difficult. I spent a lot of years living in pain, not being taken seriously. How much of that was to do with me being a woman, I'm not sure. But you can end up injuring yourself more. You can end up living with chronic pain. And if you don't have a diagnosis, you might not have access to healthcare. So understanding how to diagnose genetic disease is critically important. So because my disease mutation isn't known, it took me 13 years to be diagnosed with my genetic disease, even though I was in constant chronic pain. I was having daily dislocations, and clearly I wasn't a very healthy kid. There's no two ways about it. But the kicker is that I was only believed about my symptoms once I had a degree in genetics. And this really underlines the health inequality that exists for rare diseases. It's really difficult to be believed if no one knows how to diagnose you. So now I've touched on what it's like to be disabled in the UK and how we can diagnose genetic diseases and why it's important to diagnose genetic diseases. I'm now going to move on a little bit towards my area of research. So as you probably know, everyone watching this video today is 99.9% genetically identical. So, doesn't matter where you're from, the colour of your skin, who your parents are, where you grew up. If you're male, if you're female, we are 99.9% identical, which is pretty cool, really. But did you know that we're 90% genetically identical to a cat, or even 60% genetically identical to a banana? And this is where I'm going to start talking about my research now, because I'm actually a plant scientist, and I got into plant science because I think plants are pretty cool. You might not think the same, but I really like food, and every single bit of food in the world comes from plants. Even the meat that you eat has been fed on a diet of plants. So I think they're pretty important to understand. So plants are cheap and fast to grow in a lab environment, which is pretty useful. I can't sit around growing mice all day to experiment on. They are more genetically simple than humans, which is great, which means I know what's going on a little bit better. And we can also do genetic modification on them, so we can understand where a protein might be localized in a cell, where a gene is preferentially expressed. And I can't really sit around genetically modifying humans all day, because we all know that would break a ton of ethical and moral laws. As I said, plants are really important for food. All of our food comes from plants, even meat. And I don't know about you, but personally, I feel there's no point in curing cancer if climate change has wiped out our food system. Cure cancer all you want, but if we've not got food to put on our plates to keep our cancer patients healthy, then what are we going to be doing about that then? So, plants can undergo a lot of different stress, such as too much light, too much rain, not enough rain, high winds, plants outcompeting them. You can have bugs coming and eating them, bacteria, fungi. And what's more impressive is that when you're stressed and when you're stressed out, you can probably remove yourself from that stress. Plants can't. So it's really important to understand how plants cope with stress. And as climate change gets worse, our food supply is going to shrink. There's no two ways about it. As the climate crisis worsens, freshwater supplies reduce. So I study how plants respond to salty water and how we can genetically engineer them to grow in harsh conditions, so everyone can still eat. And the reason why salty water is important is because we don't have that much fresh water in the world. The majority of water we do have is salty or brackish, so slightly salty, and it's found in the oceans. So if we can improve plants so that they can cope with salty conditions, we have more water for our plants, and we can grow our plants in new environments near the sea. And that way we will hopefully slow down the impact that climate change might have on our food. But I can only research plants if my environment is disability friendly. So this is a look at what my workspace looks like on a day-to-day basis. You can see lots of tubes, lots of protocols, so sort of recipes for what I need to do that day, tubes full of buffers and solutions, nothing bright green and bubbling, unfortunately, but this is me from quite a few years ago now, holding a tobacco plant in one of our growth rooms. But I can only be here if my research environment is safe for me. So this is what my lab looks like, and my lovely colleague Gillian, who is trying to hide out the photo. And I've highlighted here with these red exclamation points what a normal hazard might be for someone in the lab. This is our chemical shelf. There are plenty of hazards on here. But when you're disabled, suddenly a chair becomes a hazard. That bin, if it sticks out, becomes a hazard. These drawers become a hazard to me because it might not necessarily be accessible. Even another person, if they're not properly trained, can become a hazard to a disabled researcher. And as I said, I can only research plants if my environment is disability friendly, but this has to happen everywhere in every environment too. Making work and study spaces accessible for physical and mental health needs aren't easy, but removing stigma around disclosing disability and supporting others, your peers and your colleagues is the first step towards achieving disability parity in research environments. So if you've enjoyed this mini talk, thank you so much for listening. You can find out more at Twitter, that's Emily X Armstrong, Instagram at emaarmstrong. I have a website, emilymayarmstrong.com. And if you enjoyed this talk today and fancy buying me a coffee, you can go to ko-fi.com/Emily May Armstrong. Thank you so much for listening to my talk today. If you have any questions, please get in contact and please feel free to share this video wherever you like. Thanks so much.